There you go. Now I got my volume on.
All right.
Okay.
Sweet.
Sorry about Missy. She gets very interested in what I'm doing when
That's
she's.
perfectly fine. I don't think anyone will object to having you have a cat in the video, so it's all good. All right. So, welcome back to Thought Cabin, everyone. Today I'm joined by a good friend of mine, Nick Mueller, the one who muels, and he's going to muel here today to talk about his research experience in plant science and other related fields. And then we're going to get a little bit into what you can do as a member of the public to contribute to data collection regarding ecological health and other issues like that. So, Nick, why don't you take it away and explain how you got started in this particular field? What drew you to it, and what was your first experience like?
So I actually got into plant science initially by I wound up working in an entomology lab in college.
Okay.
I didn't initially want to go into plant science. I actually started my time in university as a pre-pharmacy student. And at the time, I was just looking for jobs that would involve research. I wanted to get into research, but I didn't really know what research was. And my microbiology professor just sent out a little Canvas notification that was like, hey, this lab on campus is looking for just a student researcher who can do some work. And I saw that they worked with like plants and they worked with aphids. And I thought my plants had aphids. And I applied, and from there I've been working in research ever since.
Cool. Yeah. So let's talk about that campus experience. So, what exactly were you learning about aphids with this research project?
In this first position, so I started working in this lab in May of 2021,
Right.
and I worked there for 2 years. And in that time, I did a lot of work. I was one of several students that worked in this lab, and one of my main responsibilities was just sorting insect samples. So I was learning a lot about insects and what they looked like really up close through the microscope. And then I was also doing work like planting cotton, maintaining plants in a greenhouse.
Interesting. Is there a relation between the insect studying and the cotton studying? Were you looking at the effect of those insects on the cotton or vice versa? What's the relation there?
So I was working with a specific aphid that transmits a virus that my lab was looking at the time.
Okay.
And so we were looking for aphid counts in the field, in various cotton fields that we were working in, as well as maintaining aphid colonies in the lab. And with that, they were used for transmission experiments and such.
Right. So they were they wild aphids, or there was like a control group that had like a virus or markers on it that was released? How did that work?
So they're all one species of aphid that we just maintained on cotton. Ideally, they should not be exposed at all to any viruses.
Right.
Unless we put them on a plant with viruses.
Okay.
The aphid colony was maintained starting long before I got to that lab, so I'm not sure where exactly they got their aphids from. But to my knowledge, they're still maintaining those aphid colonies. So, yeah.
First of all, was that your first time working in a lab, like doing lab research?
That was my first time getting paid to work in a lab.
Okay.
Before that, most of my lab experience was in the classroom.
Sure. Yeah.
And so I had a little bit of experience from like previous biology labs where I was able to look at microbes and dissect things. And there was one lab that I did that I got to do a little bit of what I called like bench work. So working with some kind of more on the molecular biology side.
Okay.
But that lab was cut short because of COVID,
Right.
unfortunately. So once I got into the entomology lab, that's when I was kind of back in a lab setting.
Mm,
Though in that lab, I would say it was probably more, there was a molecular lab within there that the PhD student was working in,
okay.
but for the most part, I was working with insect specimens and plants.
Yeah, that makes sense. I know you're like a super big insect and creepy crawly enthusiast. Yeah, for those who don't know, Nick has like you've got like a snake, right, and like a tarantula and all that. Was that really like the beginning of your fascination with these creatures, or was this just kind of another nudge in that direction?
It certainly helped with
Hmm.
that.
Hmm.
Um, I definitely think that working in a lab that dealt with so many insects, I was able to learn kind of how fascinating insects are.
Sure.
I
Yeah.
I actually, after working in that lab, my insect collection did grow a lot. Though technically, arachnids are not.
Not insects. That's right.
Goodness, or arachnids aren't insects. No,
No, no, they're
it's just
certainly not.
like
Insects, see, have three segmented bodies and six limbs, whereas arachnids tend to have two segmented bodies and eight limbs.
right.
That I know.
Maybe
Yeah,
I've been out of entomology classes for too long.
you got to brush up, man. I shouldn't be the one schooling you on this one.
Right.
Okay, so your aphid experience came to an end when? How long were you working there?
So I worked in that lab until spring of 2023.
Okay. Oh,
And at that point, I transitioned into working as an undergraduate research fellow at my
okay.
university. And
Mhm.
that was when I transitioned fully into a more molecular lab. So I was at that
Uh,
point doing RNA extractions. I was doing uh, bacterial transformations. I was working with yeast a lot, trying to um, utilize certain techniques to
right.
identify protein-protein relationships.
Mhm.
And so that was a big shift away from the more ecological work that I was doing.
Yeah. It sounds like you were doing a lot of stuff there. So let's try and unpack, I guess, each of those things that you talked about. So RNA extraction, I think, was the first thing you mentioned. What are you taking RNA from and what's the purpose?
So for the I was extracting RNA from aphids,
Okay.
and with that RNA, I would do experiments to test them for the presence of viruses.
Right.
Specifically, we were looking at RNA viruses. So you can have viruses that are DNA or RNA. The virus
Mhm.
I was working with is an RNA virus, so I was able to take that RNA and then try to amplify the virus sequence from it. And if the virus was in that aphid, then it would show up on an agarose gel after I
Mhm.
amplified the sequence. So that was one
Okay.
thing I was doing with it. And then,
So forgive me for what may be a stupid question. Um, you say this is an RNA virus. Is this like a virus that has RNA proteins that you're looking for specifically in the virus, or does it affect the RNA of the aphid and you're looking for these markers that show that a change has been made?
so for an RNA virus, that means that it is a virus made of RNA nucleotides.
Okay.
So, like a DNA virus would be a strain of DNA, but the RNA
Hmm.
virus is a strain of RNA.
Gotcha.
And depending on what virus you work on, you're going to be targeting more of like the RNA side or the DNA side.
Right. Mhm.
Um, in my case, I was looking at the RNA um, of these insects. I was extracting all of the RNA from the insects and then seeing if I could amplify any viral RNA out of it.
Right. Okay. Cool. Cool. And did you? What was your result in a nutshell?
We found a lot of virus in our aphids, even though they shouldn't have had virus in them. So that project kind of went awry a little bit, where we
Mm.
we think that there could have been some contamination into our colony, but
Oh, just possibly. Yeah.
it's possible. I mean, a greenhouse isn't going to be completely insect tight, so if
Mhm.
you get one aphid
from the outside world.
the outside that has a virus that comes in and
Wow.
infects the plants that the aphids are on, then
Patient
that
zero.
starts circulating in the colony.
Mhm. So does that mean like you didn't have really a control group, or how did it affect your data?
Yeah, at that point, we had to kind of put a pause on our experiments.
Mhm.
I was trying to get, for the purpose of my experiments, I was trying to get aphids that were both negative for the virus I was looking at and positive for the virus that I
Right.
was looking at.
So you can
And
make a good
then,
comparison. Yeah.
right, but in the end, we ended up trying to collect aphids from another researcher that had a colony of the same insect. And we kind of figured it out. I was able to collect some that tested negative, some that tested positive. So
Hmm.
it worked out in the end.
That's good. Yeah, but things happen in science. At least if you can account for them, then you've learned, what you set out to learn. Let's talk about some of these other things. You mentioned bacterial studies. What was going on with bacteria? Did you shift away from viruses to bacteria, still aphids, or something else?
So I was using bacteria to clone plasmids. And so a
Okay.
plasmid is a circular piece of DNA.
Mhm.
Bacteria have them a lot of the time. They can be used for DNA, like trading kind of in bacteria, and which bacteria themselves have a circular piece of DNA.
Right.
And I was using E. coli specifically to amplify the plasmids that I had.
Mm.
So, one thing that is used often in research is it's called bacterial transformation. And you take a circular piece of DNA, a plasmid, and you can transform that into E. coli. There's various
Oh.
different methods. There's chemical methods. There's like an electric shock kind of method.
Mm.
There's E. coli is naturally competent, is what we call it, to take up plasmids. Meanwhile, some other bacteria strains or yeast strains are not naturally competent,
Right.
um, or they are not as easily made competent, if that makes sense.
Mm.
But we would transform the plasmids into the E. coli. And so as the E. coli grew and they all had this plasmid, once you get a large enough abundance of E. coli, then you can extract the plasmid from there and you have a higher concentration of plasmid that you compared to what you may have had before,
Right.
or if you just need a lot of it,
You're
it's
kind of
cheaper.
farming it.
Yeah, you're kind
Yeah.
of farming it using the E. coli. It's
Mm.
definitely cheaper than having a company synthesize a whole bunch of plasmid for you.
Sure. Sure. Now, this is the same E. coli that exists in lakes and streams and makes you sick, right? Or is it a different strain? It's more benign.
There's certain strains or specific like copyrighted, I guess, forms of E. coli. Maybe patented is the right term.
Patented, probably. Yeah.
But
Mm.
there are certain types of E. coli that are produced by companies that have certain, you can get them with certain deletions in their genes to where maybe they need a certain type of media to grow, or
Mm. Mhm.
they can have different modifications that kind of suit different research outcomes that people might want.
Okay. And then the plasmid, is that being used to synthesize like antibiotics, or what is that being used for ultimately?
So there are antibiotic resistance cassettes in the plasmids typically.
Okay.
That's how you would choose which E. coli colonies you want to use. You would
Mhm.
say once you transformed your E. coli, streak it out on a plate. Um, if your plate has a specific antibiotic that your plasmid has a resistance cassette to, only the E. coli with that plasmid can grow on that
Right.
plate.
Mhm.
And but what we were using it for is I was taking the RNA from the insect and then turning it into DNA that could then be put into these plasmids and then transformed into yeast for something called a yeast to hybrid assay. And that's
Okay.
just, it's a method that's been used for a long time, but
Mhm.
it's a method of testing protein-protein interactions.
Okay.
I don't want to get too far into that because that could be a rabbit hole. It's
Well, okay, let's talk about the yeast a little bit. What's the purpose of the yeast?
So yeast can sexually reproduce,
Okay.
and that's typically not something you would see in E. coli.
No.
But a yeast to hybrid assay is a yeast mating assay essentially,
Mm.
where you have two yeasts that have different constructs in them,
But they
and
love each other very much.
they very much do love each other. Yes. But you take these two yeasts and you have them mate in media, like a very concentrated media that's going to have a lot of nutrients that once these yeasts mate, if the two, ideally they would each have, their own unique plasmid.
Mm.
And if the pieces of DNA that we put inside of those plasmids interact, there is a signal that we should be able to see.
Okay.
And for the assay I was doing, the yeast colonies turn blue,
Ooh,
and that would be to reveal, ta-da, this looks like these two.
they're
Um,
getting it on.
they did mate, and it means that the proteins created by the DNA from
Mhm.
those plasmids likely interact or are come close enough together to interact.
Good, good. And then what's the utility or application of this research? What are
So
people
this,
doing with this data?
right. So for the yeast to hybrid assay, it is one way of trying to figure out if you have possible interactions between proteins.
Okay.
In in my case, I was looking at whether or not a insect protein and a virus protein interact. Or we know that, if an insect transmits this virus, then it's being ingested. In our case, it's a circulatory virus, so
Mm.
it would travel through the gut membrane, through the salivary glands,
Right.
and then be egested in the spit by the insect. So we're
Mm.
like, some way this virus has to move through this insect. There's something in this insect that it's interacting with. And so we would want to use the yeast to hybrid assay to kind of narrow down the proteins that it could be that it could be interacting with.
Okay.
And
Yeah.
it would just be one step. After that, there's a ton of validation you would have to do
Mm.
just to confirm, because
Right.
even though we would hope that the proteins being made by these plasmids are correct in the way that they would be in the insect.
Mm.
Yeast cells and insect cells are pretty different.
Yes.
And even though yeasts have the capability for post-transcriptional modification in ways that, say, E. coli doesn't, because
Mhm.
there could be errors in the folding of the protein, the transcribing of the protein, rearrangement could have occurred in the DNA
Mhm.
somehow. And so once you get your results, that's the beginning, and then you can start testing your results and see if you have
Yeah.
interactions.
So how do you rule out errors in brief? Are you just going to run a lot more tests, or are you studying something else? How do you figure that out?
So um, after our first mating assay and we get our first blue colonies, we would then take those colonies and streak them on a higher stringency media.
Mm.
So, say our first media that we had them growing on was a yeast media that
Okay.
was had antibiotics to kill any contaminants, and then it was a double dropout media, I believe, so it didn't have two amino acids that are necessary
Mhm.
for the yeast to grow. And the plasmids, would make up for that. They would make the amino acids in the yeast. But even then, it's possible that there's some escapes, as it's called sometimes.
Right. Things
Or
you overlooked, perhaps, or just
it the escapes would be like if the yeast can still grow on this double dropout media, even
mm.
if it doesn't really have both plasmids. And then you would put them onto a quadruple dropout media, and in that case, you would have your it'd be like if it grows and it's blue, then that's a higher probability that it is actually what you're looking for.
Right. Okay. Do you feel like by the end of your research, like you had some pretty conclusive conclusions, or was there still a lot of statistical noise by the end of it?
So yeast to hybrid assay takes quite a long time. Even though I was working in this lab on this project for several months,
Mhm.
um, I was working on this project for almost a year, I would say,
Mhm.
from prepping for the project to actually running the assays. And by the time I left, we had some preliminary results,
That's good.
but we were still confirming true positives versus false positives
Mm.
when I left.
So just like the next year, students came in and took over where you left off, kind of.
Yeah, the researcher who I left it under is still continuing that.
Sweet.
Um, yeah.
Okay. What else have you mentioned? So you've talked a little bit about work that you've done with ecological health. How do you get, how do you go from the microbiology stuff to some of the bigger stuff that you're looking at as ecological health indicators? What kind of research were you doing with that?
Is this that like citizen science?
Yeah. I'm trying to get into that kind of thing. Yeah.
Right. So I was involved with Alabama Water Watch,
Hmm.
which is through, I believe, the Alabama Extension Services. And I thought it was really cool. It's a completely volunteer group
Hmm.
that individuals go out and they test water for, they do chemical tests like hardness, alkalinity, dissolved oxygen,
Right.
pH. They also will sometimes test for E. coli levels
Hmm.
and do something called biomonitoring, where they assess stream health based on the invertebrates that are in the water. Sorry, my cat wants to play. Um, and through that, I was able to, you go out just every month to
Hmm.
streams to test for these things, and then you submit your data to the Water Watch website, and then the people who actually work for Water Watch will use that data to assess stream health over time.
Okay, so it sounds like what you're saying is you're collecting the samples and then you're also testing them. And then the data that you get from those tests, you're sending to them. So, as someone who's just kind of an individual person, not connected to any particular organization or what have you, and you go out to these streams, I'm assuming this is Alabama, like Auburn, Opelika area. Um, what are you bringing with you to test things like alkalinity or the presence of E. coli?
For sure. So I wasn't uh, doing the E. coli tests, but I was doing the
Okay.
chemical tests.
Chemical test.
There's a chemical kit that you can purchase,
Okay.
and I believe other states have similar programs that might have cheaper kits or free kits.
Mhm.
But for the Alabama Water Watch, you would have to purchase a kit. But within that, you would have all of your collection tools. So usually tall glass vials and glass bottles that you would use for the testing, but then
Mhm.
you would also have like pH indicators, and you would
Right.
have all your buffers, all of your reagents basically that you would need. And there's a booklet that teaches you exactly how to do the tests. There's trainings that they have on a pretty regular basis throughout the state that will train you on these. And you just do all your testing in the field. I would go with a friend. And so each site would take less than 20 minutes for the chemical testing.
Sure.
And you would just follow the instructions, do all your tests, and it's super easy.
Hmm.
Say for alkalinity and hardness, for those tests, there's a little like a little tablet that you would just dissolve in the water to prep it,
Okay.
and you would take a dropper and you would just swirl it around until there's a color change, and then you just record how many drops it took.
Oh yeah, like a titration experiment kind
Exactly.
of.
It'd be a
Mm.
type of titration. And then pH test would be pretty easy. Just kind of the same thing. Dissolve a tablet, hold it in, up against uh, different pH level indicator
Yeah,
color.
there's like a little color spectrum. Yeah.
Right.
Mhm.
And so they're super easy tests. I think the most difficult is like the dissolved oxygen test,
Mm.
but um the steps are super easy and they explain it very well. You collect your water samples, put in X drops of reagent A, X drops of reagent B, shake it, let it sit for a little bit, so many drops of like your third reagent, and then once your solution is clear, then you pour it into a vial and you do basically the same thing where you're counting like the volume of liquid that you add to it to make it change color. And
Okay.
so they're super easy tests.
It's a good excuse to go out with a friend and, enjoy nature for a little while, right?
For sure.
Yeah. Mhm. So, yeah, and so like, what's your kind of call to action pitch here for people who are kind of sitting around, maybe they're students, maybe they're part-time workers, what have you, and they're thinking like, oh, I wish my life was just a little bit more exciting. What's your pitch to sell this to people as like something that's worth their time?
If you want to be part of research that you don't normally get to have access to, or if you want to just be part of something bigger where you know that your work is going to contribute to, say, conserving the ecosystem or
Right.
even just monitoring stream health or the like insect identification in your area, BioBlitz type of work. You can look up online, like citizen science projects in your region.
I think what I'm going to do is on my part, I'm going to probably put some links in the description of this
Oh, good
video
idea.
for people to probably like do a generic search, and maybe I'll have some specific links for like the Mid-Atlantic or the Deep South region and things like that.
Right.
Um, yeah, would you say that in your own personal experience with the research you've done, have you found anything else about the local ecological health that surprised you or alarmed you or pleased you? When you're doing these tests?
Sorry.
All good.
Missy.
Bring
She's
the cat
very
up. Bring her up here. There's Missy.
hey, this is Missy. She,
Oh goodness.
she
She
really doesn't like to be on camera.
camera shy, huh?
Yeah. Every time she's doing something cute and I point a camera at her, she stops doing
She
it.
stops doing it.
Yeah.
She doesn't exist when she's being observed. The opposite of the
Mm.
universe, right? Um, anyway, so like I was saying, yeah, the research that you were doing in your local area, did any of it surprise, alarm, or please you? What was the result looking like?
I thought for the project that I was working on for Alabama Water Watch, yeah, I thought it was really cool. Every month you go out to the exact same spot
Mhm.
and you can kind of monitor over time how things change. We
Yeah,
noticed when it got colder, our results would be different from when it's warmer outside. And like our dissolved oxygen would be different, our pH would be different. For our biomonitoring, where we were identifying different macroinvertebrates in the area,
mhm.
we found that that would change over time. Some months we would see like a whole bunch of crawdads in the water. Some months you would see a ton of naiads, which are dragonfly nymphs.
Right.
And you can kind of expect different things for different locations, and you can notice if for some reason your measurement from one location is way off than what was say the month prior or how it usually is.
Mhm.
And you can look around and oftentimes you'll find that there's new construction in the area that's
Right.
causing runoff that is impacting the health of your streams,
Mhm.
or you might find that there could be a big change in water level. One of our sites actually, our first like 2 or 3 months doing it, the water was super deep because we were working. There was like a bridge road
Mhm.
kind of area, and a lot of the path for the water to flow under was blocked by debris from trees and
Oh yeah.
leaves and stuff. And so the water level was really high. And then we showed up one month and all of that debris was clear. So now I'm like, our water level's like only a few inches deep. What
Play
happened?
strained, huh?
Yeah. And
Gotcha.
so it was it's kind of cool seeing those areas kind of change over time, and you can see how different conditions kind of impact the water in different ways.
Well, to those of you in the audience who think that this sounds like a worthwhile endeavor, I think it very much is. And we're going to go out in a little bit, and I'm going to show, what this kind of data collection looks like. Nick, of course, thank you for being on here and talking about all this stuff. And I do hope that those of you who are watching are inspired by this, and that you want to do your part to contribute to the general body of knowledge that we have in science, whether it's ecological health or it's looking at antibiotic resistance or anything else. We need people who are hungry for knowledge about the world to go out there and collect that data, perform those experiments, verify those results, and just make sure that we know what the hell is going on in this world, because it's a crazy time. Yeah. Thank you.
Sweet. Thank you for having me, Daniel.
Of course.
Okay, so I'm currently about a half-mile away from my house, standing outside of a stream that feeds into the Patapsco River. The water level right now is kind of low because it's been a few days since it rained, but this is moving water. It's not stagnant, so it should provide us a pretty decent idea of what the ecological health of this area is right now. So what I'm going to do is I'm going to go down to the surface and I'm going to collect some water into this little Tupperware thing. And then I've got a testing strip that I'm going to test the water with later. Good stuff. Okay, so right here what I've got are some 5-in-1 easy strips. These actually are for fish tanks, technically, but I can use them for this particular application as well. So what I've got right here is this strip, which has 5 little chemical elements on it. And each of those chemical elements represents a different marker of water health. So that includes, according to the box, this strip will allow me to test for nitrate, hardness, pH, nitrite, and alkalinity. So, just simply going to submerge right into the water. And then in a little bit, we're gonna compare and look at how these colors have changed. Okay, so it's been about 30 seconds of water contact with these chemicals, and we can see some clear results. So over on the box right here, we see the colors that these things are supposed to be. And then over on the left, we see what colors they actually are. So the nitrate and nitrite levels are basically within the normal range. It's about what we expect. But hardness is supposed to be green, and it's coming out as orange. Alkalinity is supposed to be yellow, and it's coming out as kind of a teal blue. And then finally, pH is supposed to be kind of a yellowish orange, but it's coming out as a salmon pink. So that's telling us that there's a few different markers that just really aren't quite where they're supposed to be. Some possible explanations for these anomalies really boil down to just kind of understanding where we are locally. There's a lot of runoff from the local suburbs, and people are going to be using things like detergents, car cleaners, pretty much anything you might think of. Even in some places, it's going to be pesticides and other things that might get sprayed onto farmland, and fertilizers, and all these things tend to impact the alkalinity, hardness, and pH of the local water. As for whether or not it's still healthy enough for creatures to live, that's a matter that probably requires further observations. And now I'm going to return the sample to the environment that I collected it from. I hope you found this video informative and entertaining. But most of all, I hope it inspired you to consider being a citizen scientist yourself. It's very easy to go out there to any local reservoir and collect data that scientists can use to figure out what the health of the environment is. If we all work together, we can learn more about our planet, and we can figure out what needs to be done to help it. Thanks for watching.